Good morning, biology students. This video is to take place of the class today that Mrs. Horton missed due to internet connection issues. So I do apologize, but hopefully you'll find this video useful. In addition to that, I'll be sending out some videos for Thursday and Friday because I won't be here, as we have, all teachers have professional development. So you won't see us. I will be available through Skype, hopefully in the evenings. So if you get stuck on something, just Skype me. Even if I am not online, I will respond later. In addition to that, you can email me, you can text me as well. You can find that information in the contact me. Use the recording list. You can use the old recording list as well if you see things that might be helpful to what you're studying from. So if you want to do really well on the final exam, I would start preparing now, just going back through your notes a little bit every day until our final, and our final opens up on the 10th. I was really happy. Mrs. Cunningham gave us permission to open it up on the 10th. So that is next Friday, not this Friday, but next Friday. And then the 15th is our last day of school. That's not much time for those of you who are behind. I have been really pleased with our class, though. Everybody in our class, and this is the first time I've ever had this happen since I've been teaching at K-12, everyone in this class is working. Everyone in this class took their Math and English USA test prep. So you guys really need to pat yourselves on the back. I've had so many people coming to class and so many people coming to Science Help and communicating with me. So you guys are doing a great job, so keep it up. Just a reminder, the labs and the unit and mid-unit assessments are worth so many points. You cannot pass this class unless you do the labs, and that includes the discussions that are associated with those labs. Those labs add up to more than 60% of your grade. So just keep that in mind. The quizzes don't count for that much. They're just practice getting you prepared for those labs and for the assessments. In addition, you should be doing the USA test prep daily. Next week, there will be 4 USA test prep assignments that actually will be for points. Now, I don't grade them based on what you get on them. They are practice for the AIM Science. So if you complete everything within that practice, you will get 10 points, and that's worth 40 points overall because there's 4 of them. I think that's it for announcements. I want to tell you guys I've really enjoyed this class, and I know you guys are going to do great things, and I hope that you stay with K-12 and that next year I might see some of you in environmental science or in chemistry. Mrs. Poling teaches chemistry, so some of you might, hopefully all of you will want to go into that, but I know some of you will go the other route, which is not the university route, so the technical college route, in which case you don't have to take chemistry unless you're really interested in it. Which I have lots of students, I think, that in this class in particular, that I think will just fly through that class and do wonderful work. So next week we'll have some practice on essay questions for the final exam. Monday we'll go maybe review a little bit for the unit 4, the ecology unit exam, and we'll see. We'll figure it all out. We have reteach next week. We have reteach this afternoon, and you can make appointments. I'm going to try to have some extra classrooms open for questions about labs and redoing assignments and things like that. If you need a catch-up plan, my suggestion for those who are somewhat behind, go to your grade book, see what assignments are there, and find the associated unit. Now, the number on the lesson, the 4, well, one is associated with molecular genetics. 2 is associated with the evolution unit. 3 is associated with the human systems unit. 4 is associated with the ecology unit. Now, there's an extra unit in there, and there's a quiz that's not labeled with a number called bacteria and virus. So bacteria and virus go into the human systems unit because it's kind of a review of what you learned or were supposed to learn in the curriculum for Biology A. So we kind of went over those two. I would review your fern reproduction, annelids reproduction, and you can find that in the extra reading that I gave you and also your lessons. I didn't really go over the annelids very well, and I will try to find a recording to support that because I do believe there is a couple questions on the unit exam about that, but we can talk more about that on Monday. I'm going to do our lesson now, which is the going over nitrogen fixation. So it covers part of the nitrogen fixation or the nitrogen cycle. And I'm going to give instructions on the lab that you guys are going to be starting tomorrow since we don't have our lab day. So it's kind of a long recording and I do apologize. So let's go ahead and get started. Let me move over to screen 2. Okay, hopefully all of you can see that now. So 4.15, you guys all should have been growing your plants, so your bean plants and your radish plants. And this lab is where we actually are going to dissect those. But first, we're going to go over the lesson that's associated with it. Here is our Success Happens Here slide, no exceptions. Oh, let me go back one. And then also Kids at Hope pledge. I am a kid at Hope. I'm talented, smart, and capable of success. I have dreams for my future, and I will climb to reach those goals and dreams. This session will be recorded for learning purposes, and learning purposes include a lesson review for students who are absent, students who want to review for a test, etc. The objectives for this lesson come from our Arizona Science Standards, and there's 2 of them. So Strand 4, Concept 3, and Concept 5, PO3. So if you are interested in that, you can go take a look on the Arizona Department of Education's website if your parents or your learning coaches are interested as well. So these concepts that we're going over are concepts that you might see a question on AIMS about. You will be able to, at the end of this lesson, you should be able to describe in several sentences how bacteria and nitrogen are important to plants. And you'll need to be able to use the vocabulary words that we learn and talk about in this lesson. Your bell ringer, I just want you to think about the nutrients that plants need. So go ahead and stop the video for a second, think about it, and then as we go through the lesson, you'll see more about this. Ecosystem and the nitrogen cycle. So the nitrogen cycle describes how nitrogen moves between plants, animals, and bacteria, the atmosphere, and the soil in the ground. So remember, those are our producers and our consumers and our decomposers. So they're all impacted in the food chain by this nitrogen cycle. So nitrogen is an important element to all life on Earth. And there are different nitrogen states. So we know that nitrogen on the periodic table, we look at it as N, right? Well, it's considered one of the organic molecules because organisms use nitrogen. They need nitrogen. And for nitrogen, in order for nitrogen to be used by different life forms on Earth, they must change into different states. Nitrogen in the atmosphere as a gas molecule is in 2. That means that there are 2 nitrogen molecules attached to each other. Other important states include nitrates, which are nitrogen and oxygen. So 3 oxygen attached to 1 nitrogen. And then we have nitrites. So notice the difference. There's an I, not an A, nitrites. And that is 2 oxygens attached to a nitrogen. And then there is ammonium, and that is NH4. That means 4 hydrogen attached to a nitrogen to create the ammonium molecule. So those are the different states, that the 4 important states that nitrogen can be in. The picture below shows the flow of the nitrogen cycle. So like the water cycle, there's a nitrogen cycle. And the most important part of that cycle for plants and humans really is bacteria, the rhizobium. So when you planted your plant, your bean and your radish plants, you had a package of rhizobium bacteria, and you actually put that rhizobium into the soil. And it's so important, and that's what helps the bean plant grow. Bacteria help the nitrogen change between states so it can be used. In the nitrogen-fixing plants cannot utilize the nitrogen in a gas form, and so this bacteria helps them. When nitrogen is absorbed by the soil, different bacteria help to change the state so it can be absorbed by plants. Animals then get their nitrogen from the plants. So the plants that we eat, the animals that we eat, we get nitrogen from that. The close relationship between the bacteria and the plant is called symbiosis. And that is a close interaction between organisms of different species living in contact with one another. And we're going to talk a little bit more about what that looks like for our bean plant. There are a few processes in the nitrogen cycle that we need to understand, and then we'll go back and look at the picture one more time after we define these. So we have fixation, and fixation is the first step in the process of making nitrogen usable by plants. And here, bacteria change nitrogen into ammonium. So let's go back and look at fixation. So we see nitrogen-fixing bacteria living in the legume root nodules here. We have atmospheric nitrogen at the top, plants with assimilation. We have denitrifying bacteria and nitrifying bacteria. So there's our nitrates and our nitrates and our nitrites. We have nitrification, ammonification. So these are all the different things that are happening, but fixation is occurring right here with the nitrogen-fixing bacteria that lives in the legume root nodule. So right there. Let's go back to our definitions. Nitrification, this is the process by which ammonium, so NH4, gets changed into nitrates, that's NH3, by the bacteria. Nitrites are what the plants can then absorb. Or I'm sorry, nitrates. So NO3, nitrates is what the plants can absorb. Assimilation, this is how plants get nitrogen. They absorb nitrates from the soil into their roots. So there are some plants that don't have the symbiotic relationship, but they are able to absorb that those nitrates into their roots. Then the nitrogen gets used in the amino acids, in nucleic acids, and in chlorophyll. So remember, amino acids are the building blocks for proteins. Nucleic acids are the building blocks for DNA. And chlorophyll is what is used in photosynthesis, and that's where photosynthesis actually occurs, within that chlorophyll in the chloroplast of a plant cell. So those three things are super important, and plants could not, and animals could not function without the assimilation of nitrogen. So ammonification, this is part of the decaying process. So when a plant or an animal dies and decomposes, like fungi and bacteria and worms, they turn the nitrogen back into ammonium. That's NH4. So it can re-enter the nitrogen cycle. So there are bacteria that like that NH4. They break it down and add oxygen back into it, so that it can be taken up into the roots. Denitrification, so that's this bottom one, is extra nitrogen in the soil and it gets put back into the air. There are special bacteria that perform this task as well. So let's take a look at that picture again. Hopefully it'll make more sense. So we do have the plants that go through assimilation right here. We do have plants that use the nitrogen-fixing bacteria that live on the roots here, which is our bean plant. Our radish plant practices assimilation. We have bacteria in the soil that are breaking down the organic material to form NH4 ammonium. And then we have bacteria that break that NH4 down into nitrates. We have bacteria that actually is called nitrogen-fixing bacteria that takes that NH4, brings it up to the plant, and breaks it down, and that's what the plant's using. So the nitrification happens when with the breakdown of the ammonium into nitrites. And then from nitrites, there is nitrifying bacteria that adds in another oxygen into the nitrates so that the plants can assimilate it. So it just is a complete circle. It goes around and around and around and continues with the cycle of life. Oh, let me go back to that picture again. So when an organism dies, like this rabbit or a plant die, and that fungi and the bacteria and the different types of worms and bugs start breaking that organic material down, it's breaking down those amino acids and the nucleic acids and the chlorophyll. So all those are being broken down into a simpler form. And that's where the nitrogen comes from when it goes into the soil. Why is nitrogen important to life? Well, plants and animals could not live without nitrogen. It is an important part of the cells and processes such as amino acids, proteins, and even our DNA. It is also needed to make chlorophyll in plants, which plants use in photosynthesis to make their food and energy. And that is why nitrogen is so important to life. How have humans altered the nitrogen cycle? Now, this is, I have to just put it out there. There are two schools of thought on global warming. Just going to use that political word. I don't like it because it's a political word. It's not really a scientific word. We as humans have activities that can impact the nitrogen cycle. So if we brought it down into like our garden, and maybe a plot of land that has plants on it, but it's not our garden, it could actually be impacted by us because we put synthetic fertilizer on there or just too much fertilizer. Fertilizer holds lots of nitrogen. So those are things that, you know, we have to think about when we are wondering why that nitrogen cycle is disrupted because we altered it in some way. So human activity has altered that cycle. We do this by adding that nitrogen into the soil with fertilizer, as well as other activities that put more nitrous oxide gas into the atmosphere. This adds in more nitrogen than is needed by normal cycle and upsets the cycle's balance. So it could actually change the level of bacteria that's in a garden, which is one of the reasons why gardeners like to use compost, because it's a natural way of decomposition and putting that decomposed material into your soil. It has the bacteria, it has the things that we need in order to make our gardens organic. Agriculture farmers use it as well, that they can use compost, they can use natural fertilizers and not synthetic fertilizers. Fun facts about nitrogen: about 78% of our atmosphere is actually nitrogen. However, this is mostly not usable by animals and plants because it's in a gas form. Nitrogen is used in fertilizer to help plants grow faster. Nitrous oxide is a greenhouse gas. Too much of it can also cause acid rain, which changes the pH level of the soil, which can affect seed germination. So that was one of the labs that we just finished up. One of the plants did great in a higher pH level than the other plant. It liked a more alkaline or neutral pH. Nitrogen has no color, no odor, and no taste. So you don't know that it's there. About 3% of your body weight is nitrogen. So that's kind of crazy. So like if you weighed 100 pounds, then 3 pounds of that would be nitrogen in your body. Oh, let me go back to this slide real fast. Another fun fact, another way to cause nitrates to be formed is through electricity. So, for example, if you have a bolt of lightning hit the ground, then it can cause nitrification with the gas nitrogen in the environment. So it puts it into the soil. So that's another way that it gets there. Nitrogen-fixing bacteria form that symbiotic relationship with plants, but not all plants, just some, like legumes, like the bean plant. Nitrogen-fixing species of bacteria may be free-living in the soil or may inhabit cells of the organisms that benefit from their nitrogen-charging ability. Bean plants and many other members of the legume family and other families of plants form these symbiotic relationships with bacteria. Relationships visually evident by the presence of a root nodule. So when you dissect your plant during this lab, you're going to see they're white nodules, and they're pretty. With your plant, they're probably not going to be very big because the root system isn't as well established as some. But you'll see them, they're little round white nodules on the roots. They're very fragile. So Rhizobium form symbiotic relationships with some plant roots. And then on those roots, they form nodules, and those nodules help with fixing nitrogen into the root system. And in your lesson, make sure and read that, your lab lesson, because it'll talk more about how this actually happens. So the goals for your lab are to analyze root nodules. So you're going to look at those, and you're going to relate their structures to their function. And we'll talk, like I said, I want to review this lab a little bit next Monday. So we'll talk a little bit about the structure. We can look at the structure and we can talk about what your results were. And then we want to be able to relate the nitrogen cycle to the presence of root nodules in some plants. So we're going to compare the radish plant to the bean plant. So this plant right here is actually a legume. It isn't a bean plant. The one on the far right here is a bean plant, not planted in soil, but you can see kind of what it looks like and the roots in the bottom. The radish plant does not have nodules, so you'll look at the roots and see that in comparison to the bean plant. So for your lab, you're going to need a few things. You're going to need the plants that you planted, so the radish and bean, student grown. Takes about 8 weeks, so if you haven't started that, it's too late. If you haven't started your plants, you can buy them from a garden store. You just have to go and ask for a nitrogen fixing plant, so one that has nitrogen fixing nodules on it. And then you can also, from the grocery store, buy your radishes as long as they have the root on them. So you just have to ask for a radish with the root. Or you can ask the garden store to for a root plant, so one that's like a radish. They might have one there. In addition to that, you'll need newspaper, so down here in red, newspaper. Those two things are not in your lab kit. Everything else is in your lab kit. You're going to need your forceps. Those are the tweezers, pipettes, and plastic pipettes. You're going to need a few of those. Gloves, disposable. Those have to be your disposable gloves, the blue ones, if you have any left. If not, we're not really working with anything that's going to harm you. It just protects you from the dirt. So just keep that in mind. And it also makes it so that you're not putting any extra bacteria on the microscope slides when you create them. You're going to need your goggles for safety, and microscope, so compound microscope, the light microscope that everyone should have received. You're going to need your scalpel or a knife if your scalpel is completely dull. Just be very careful with that. You'll need your apron, your hand lens, so that's that little tiny hand lens that's in a red box that's hard to get out. You're going to use that to look at the roots. You'll need your microscope slide cover slips. So you'll need the microscope slides and the cover slips because you're going to be creating slides to look at under the microscope from your roots. There's also a prepared microscope of the Rhizobium nodules, so a legume nodule with Rhizobium, and you're going to want to look at that and sketch it. And you're going to want to sketch whatever you see under your hand lens, whatever you see under the microscope. You're going to want to sketch it and label it. You can scan those in, you can take a picture of it, you can draw it using drawisland.com. So www.drawisland.com. I've given that URL to you guys before. But those sketches do need to be turned in. So that is proof that you did this lab. And this one is really important because this is our lab, our last big lab. And if you're going off to university, they may require you to show through pictures that you actually did the lab since you are doing school at home or online. Forms to download are your student guide, so 4.15. You'll also want to look at lab safety and we're not dealing with any chemicals, so you shouldn't have any issues with that. So you don't need the MSDI sheet. You do need the graded assignment, which is in 4.17, I think. Oh, maybe not. Maybe it is in 4.15. So graded assignment called Fixation in Root Nodules. So when you first open the lesson down at the bottom where the folder to turn the assignment in, there's digital material there, and that's where you download it from. So let's look at our lab procedures. You're going to be observing the roots. So each time you need to draw on the lab report, carefully remove your gloves and wash your hands. Put on your gloves before handling the plants or the roots again. And that's just because the bacteria on your hands can actually contaminate what you're going to be looking at later under the microscope. So it's really important that when you're handling the plant that you have your gloves on. First inspect the bean and the radish plants that you have been growing for the last few weeks and choose the cups with the largest plants. So the largest bean plant and the largest radish plant. You may dispose of the others or plant them outside if the season is right for growing vegetables. So you can, so you could actually put the other plants into a larger pot even to continue growing them. Let's see, cover the work area with newspaper. You can use plastic or wax paper if you have that. I've used plastic wrap before, but I don't suggest it because it tears. And place the cups with the largest plants on it. Gently grasp the base of the bean plant stem and carefully pull the plant out of the cup. Note, it may be helpful to hold the cup sideways or upside down over the newspaper. So if the plant, one thing that I've done before is I kind of tear the styrofoam away from the plant, away from the soil. So I kind of open up one side and then you can look at it that way to start with. That'll give you a good view of it where you haven't disturbed the root system. So you might want to do that first before you completely pull it out. Gently shake off or wipe away any excess soil from the roots so that they are clearly visible. Be very careful when you're doing this, gently, very gently, because the root system is fragile. Observe the roots of the plant and then record your observations, including a sketch in the data chart on the lab report. So you can create your sketch, like I said, and draw island. If you are not able to take a photo of your sketch or upload it through scanning, you can do it that way. And then, so that was for the bean plant, and for the radish plant, you're going to just repeat those steps. So next, you'll be observing the rhizobium with the hand lens. So you're going to use that little hand lens that was in the red box. And remember that when you're done observing, you need to take your gloves off to draw your sketch. So you're going to use the scalpel to cut a large nodule from the bean root. So find a large one. They're not all that large at this point. If you were growing for more than 8 weeks, they might be larger. You're going to cut the nodule in half with the scalpel. Okay, so if your nodules are too small, then I want you to go online and find a picture of a nodule that's been cut in half. And you're going to use that to sketch. But I still want you to use the hand lens to view that nodule and try to cut it in half and sketch what you see inside. And you can go online and find information about that nodule and label it from that point. Next, you're going to get a blank microscope slide and cover slip from your lab kit. Check to make sure that both are clean. If not, you need to wipe them with a paper towel or clean cloth. Use the scalpel to cut off a thin piece of the nodule that will fit under the cover slide. Use the forceps to put the nodule slice onto the slide, and you want it as thin as possible, because if it's too thick, the light won't go through it and you won't be able to see all the different cool parts. So using a pipette, add a drop of water to the slide, just one drop. Then, using the forceps, gently place the cover slip on the nodule slice. Place one side of the slip down first and then slowly let go, letting the slip fall gently onto the drop of water. Avoid trapping air under the slip. If you see air bubbles, remove the slip. Clean the slide and repeat steps 3 through 5. Gently press down on the cover slip with your thumb to squash the sample, but you don't want to do it too hard. To reduce possibility of breaking the slide, be sure to do this step on a very flat, hard surface. View your slide with the microscope and then sketch what you see on your lab report. So you should have, this will be the third sketch. Then view the prepared slide from Key 12 Materials Kit and compare it with your slide. If your nodule does host ribosobium, it should look much like the prepared slide. The prepared slide has been stained with a dye that attached itself to the ribosobium. The dark reddish purple areas on the prepared slide are those with the highest concentration of bacteria. Now, you could do a little research and find out if ribosobium is positive or negative, and then go look to see if you have any materials or any dye, because you did a lab, I believe in Biology A, that you used dye with your microscope, and you can dye it using the methods that you guys learned in the beginning, and see which one you can actually dyes the bacteria. So that's a little experiment that you can do making your slides. So when you've finished, clean the slide and cover slips, rinse the scalpel and the forceps and the pipette, and wash your hands thoroughly. Make sure and clean up the newspaper and your whole area. While you're doing this slide, please make sure that young children and animals such as cats and dogs can't access it while you're working on it. I do know that bean plants, if they have seeded, are edible, so just keep that in mind. Radish leaves are as well, but don't eat them because this is your lab. Ticket out the door is really why ribosobium is, wow, my grammar's bad, why ribosobium is so important to the bean plants. And, you know, you need to be able to reference the nitrogen cycle and the different types of nitrogen fixing activities that are going on in the soil that help bean plants. And remember that you can also mention radish plants because they use a different portion of the nitrogen cycle than the bean plant does. So that is it for our lesson today. Please make sure and go through everything that, you know, all your reading. You know, you might have to do a little bit of homework as we go through the next couple weeks, beyond your 1.5 hour each day. Since you're taking 4 classes, it ends up being an hour and a half each day for biology. Like brick and mortar, you know, you might have a little bit of extra homework, and that really is the practice, the USA test prep. That should be your extra homework over and beyond what you're doing with the assignments that I give you. So I actually measure them out in average time, you know, what the average student does for length of time in the lessons. And that's how I decide what lessons to give you guys. So just keep that in mind. The next 2 weeks, you're probably going to have a little bit of homework because of the practice. I've noticed in our class, though, a lot of people are not doing the USA test practice. Please do it. It will help you so much. And we are going to be looking at those in lessons, your personal ones. You'll be looking at them in our lesson, and we'll be discussing them. There will be activities because I really want to prepare you guys so you're not surprised when you take that AIMS science test. I want it to be a breeze for you. Let's see. I think that's it. So you guys have an awesome day, and I will see you back on Monday.